So we will first hear from Tijl Grootswagers from Western Sydney University, and he's going to be talking about the neural dynamics underlying prioritization of task-relevant information. So I will ask the other speakers to please turn off your video and microphone, and Tijl, you can go ahead and share your screen. Looks good to me. Okay, whenever you're ready, take it away.
Thank you, Megan, and thanks everyone for joining us in your late evening, early mornings, middle of the nights. I'm fairly comfortable here at lunchtime in Sydney. My name is Tijl Grootswagers, and I'm going to talk to you about a recent study of ours. And it's about information prioritization. And there's a lot going on in the world around us. And if we are not prioritizing the right incoming information, this has obvious consequences. So this is a fascinating topic that has seen a lot of attention, pun intended, over the decades. This question about at what neurostage and at what time in the brain's response to incoming information is task-relevant information prioritized. So I'm going to use task-relevant and attention a bit interchangeably. And we know a lot about this. We know about things that are presented in specific locations, at what time points those are enhanced. We know that certain features can be amplified and enhanced in neural responses. And in streams of incoming information, we know that relevant targets in these streams are see a very specific enhancement around 270 milliseconds. So there's a lot of research on this topic already, but what this often has in common is that the stimuli are presented in isolation. So it's often a cue, one stimulus, and you do something with the stimulus. And we were wondering what about stimuli that are presented in the same location and at the same time? So when there's a lot of competition going on. So what about stimuli that overlap in space and time? So that's what we focused on in this study. So basically the question is how attentional prioritization works when stimuli occupy the same space and are presented at the same time, and specifically which processing stages, so how does the time course of this, how is the time course affected by attention here? And we use so this is generally very difficult to disentangle, because when you present two stimuli at the same time or very close in time at the same location, it's very unclear which signal in your neuroimaging response belongs to which stimulus. And we've shown in a couple of papers last year that when you use so fast streams of information, so RSVP, in combination with EEG recordings and MVPA, so classification, you can actually disentangle the overlapping information that is specific to each of these stimuli in the stream. So we've got a couple of publications from last year that really nicely show this. So for this current study, we're doing the same thing. So we're presenting stimuli in relatively fast streams, and we're recording EEG, and we're using classifiers to figure out what information belongs to which stimulus and how this changes depending on task demand. So here are stimuli. We use 16 different stimuli. There's some birds, fish, boats, and planes, but generally we're interested in the decoding of the individual stimulus in the sequence. So we present these in relatively quick sequences. So these sequences are at 2.5 Hertz, and they're on. So each stimulus is on for 200 milliseconds and then gets taken off the screen for 200 milliseconds, and then we have the next stimulus coming on. And we asked subjects to do a two-back detection task on the stimuli. And to make it even more complicated, there's two stimuli on the screen. So one is the objects that I was just showing you, so those 16 images, and we presented them overlaid with letter stimuli. So we had 16 letters, and they were presented at the center of the object on the screen. And we asked subjects in half of the sequences to respond to two-back events on the object stimuli. So here that would be the boat is repeating in a two-back event, so then they would press a button to report this. In the other half of the sequences, which were actually yoked, as in we've shown the exact same sequence, but we asked subjects to respond to the letter stimuli and two-back events on the letters. So here they would not press the button whenever the boat was repeating, but they would press a button when that letter U repeated to stimuli after. Now, as you may notice, there's a co-varying feature in this display, as in the letters are always the small element on the screen compared to the objects, which are presented at a much larger eccentricity. So to look at the interaction effect here of the relative size between the stimuli, we repeated this exact same experiment in a new set of subjects where we switched the location of the two stimuli. So now we have small objects overlaid on large letters. So this allows us to really nicely disentangle these effects. So they were doing the exact same task when we presented the stimuli in the exact same order as well, but they are different subjects. So to illustrate this, I'm going to show you the two experiments, sort of to give you an idea of how fast this is going. This is a pretty difficult task, and people can do this task, but you find that they are, for example, they were not responding, they were not giving any false positives on the unattended stimulus. So if they were asked to do objects, they would not give you any letter 2 backs. So this tells us that the manipulation works really well, right? So we can direct their attention to one set of the display really easily in this case. So we have 20 participants in each experiment, and we did minimal EEG processing as we've done in our previous work as well. So we do some filtering between 0.1 and 100 Hertz, downsample the signal to 250 Hertz, and then create epochs relative to the onset of each individual stimulus in the sequence. And these epochs lasted 100 milliseconds before stimulus onset up to 1,000 milliseconds after. So there's actually multiple events in this sequence, but we can align them to the onset of each individual stimulus. And then we use MVPA to decode the overlapping responses to the individual stimuli. So the first thing that we did was discard all the 2-back target events. So we do not actually look at the target processing. So we were mostly interested in how does attention prioritize the information, all the stimuli in the sequence. And all the stimuli could have been targets, but were not in this case. So we discarded 2-back targets. So all the stimuli are still relevant. And we use a leave one sequence out cross-validation. So we train classifiers on all but one sequence, and we test the classifier on the remaining sequence. And we train the classifier on distinguishing between the 16 objects, between all pairwise combinations there, but also between all the 16 letters that we used. And then we repeat this process separately for object tasks and letter tasks. So when they were responding to object 2-backs and for letter 2-backs. And then we repeat this also over all the time points in the EEG epoch. So we get a decoding accuracy over time relative to the onset of the stimulus. So here's the results for object decoding. So here we're decoding pairwise objects. So chance level is 50%. And we see really nicely that the classifier is indeed a chance before the stimulus comes on. So that's our sanity check. And then we see that decoding accuracy shoots up to around almost 60% accurate. For both tasks, so regardless of whether people were responding to objects or for letters, we see that decoding accuracy increases. And we see that the lines start to diverge around 200 milliseconds here between the different tasks. So in blue, we have the object task, so when the objects were attended, and in the dashed orange line, we have the letter task, so where the objects were not attended, but we could still decode the objects. Here are the associated topographical maps. So we see that the signal originated from occipital senses. And here are associated statistics. So here I put a blue dot in each time point whenever the decoding accuracy was significantly above chance. I also use Bayes factors in this case. So there's a blue dot in the second row whenever the Bayes factor was showing evidence for above chance decoding, and there's a blue dot in the bottom row when the Bayes factor was showing evidence for the null, so no decodable information. And in blue, we have the attended, in orange, we have the unattended, and in black, we have the difference. So we see that the difference starts to come on just after 200 milliseconds. So this is when the object was large and the letter was small. And in the second experiment, we swapped these two displays around. So I'll put them side by side here. And we see a very similar picture here, except for the very strong first response. So that strong early peak is gone. But we still see a difference coming on around 200 milliseconds. And in fact, it looks like it starts a little bit earlier when the object was the small item on the screen. So we see an enhancement of task relevant information in this case around 200 milliseconds, but it seems like that was happening earlier when the object was a small stimulus. Now, we also decoded the letter stimuli in these sequences relative to their task. So here's the results for letters. So I think the first interesting thing is that, yes, we could decode between individual letters. It looks slightly worse than for the objects, but it was still possible. But letters are a bit less complex stimuli. So this is, I think, why it's a little bit lower. And well, we see some interesting effects here. So we see an earlier difference for letters whenever the letter stimulus was small. So this difference started around 100 milliseconds compared to the objects around 200 milliseconds. And when the letter was large, we again see that the difference seems to start a little later. So we see an early low-level attentional enhancement for letter stimuli when they were small on the screen. And the effect was stronger earlier when the letters were small. So in conclusion, what we found that regardless of the tasks that people were doing, early neural responses to stimuli in these fast changing streams display remarkable detail about the sensory environment. So we could decode between individual objects, we could decode between letters, regardless of whether they were attended or not. And the effects of attention seem to interact in a manner dependent on the perceptual characteristics of the display. So whether the object was small or large and whether the letters were a small or large item on the screen. So with that, I would like to thank all the people involved in this project, and I will point you to the relevant preprint that summarizes this research on bioRxiv, and I would like to thank you for your attention.
Great, thank you so much. And in lieu of applause, because we can't do that in Zoom, I'd like to invite the audience to drop some praise into the chat if you enjoyed the talk. So thanks for the great talk. If you do have questions for Tijl, please drop them in the Q&A box as well, or you can put them in the chat. I'll see them either way. But while we're waiting for some of those questions to come in, I was just wondering if you might tell us a little bit more about whether you're doing so you have this decoding performance that's kind of comparable, at least between the small and the large, and also between like the letters and the objects. Do you see though differences in, I don't remember whether you said what kind of decoder you're using, but differences in the informativeness of the features in terms of the anatomical location of like which electrodes are really strongly contributing? I don't know if you've done any statistical analysis on that yet.
We don't have any, that's a good question. We don't have any statistical analysis on this because it really becomes, it explodes in terms of complexity as soon as you start looking at individual electrodes and at individual time points. But sure. We do talk a little bit about different. It seems like it's a complex story. So that it looks like there might be differences in the type of lateralization that you see between these two effects. We talk a little bit more about this in the preprint, but there's no strong, reliable differences that we concluded from this.
Great. Cool. Okay. Well, if you do have lingering questions for Tijl, please do drop them in the Q&A, and I'm sure he can answer them in chat on the back side. But if there's no other questions, I guess